see the power of cohort studies. That was fantastic. Thank you so very much. Our next speaker, Dr. Semir Beyaz, is based at Cold Spring Harbor Laboratories. And so we're very excited that that means he also has an association with Northwell. And Semir is going to talk to us about the diet, microbiome, immunology, and cancer risk. So back to the lab, back to the bench, to try and understand what is going on. Semir?
All right. Well, thank you so much for the organizers to invite me, and thank you all for being here, for all the great work that you have been doing, because my interest as a scientist in colorectal cancer started a little bit serendipitously. I was an immunology PhD student at Harvard working on epigenetic mechanisms in immune cell development. And then I realized that serendipitously I have a tubular adenoma, a small polyp. And then I did all the testing that you guys described from the clinic, and it was all negative. So that was an interesting scientific question for me to get a second interest and start understanding how environmental perturbation may influence cellular states that can increase risk of cancer. So today I'm going to describe to you some of the previous studies and some of the ongoing studies that we are working on at Cold Spring Harbor Laboratory. So my lab is interested in understanding a very simple, fundamental, seemingly simple, fundamental question, whether or not the food, the nutrients that we take through our body influence disease or health states. So this is a very complicated problem because what you eat is always incorporated into 1,000 dimensions when it gets into your body, and it's very difficult to make causal mechanistic conclusions. So, coming to Cold Spring Harbor, I decided that I will take this question as my initial question. And one model that we use as a perturbation in response to some nutritional link is obesity. So obesity, as the speaker before me nicely demonstrated, it has been rising in the US and also in the world. And this is not just limited to adult obesity rates. Recent reports suggesting that childhood, adolescents, and adult obesity rates are increasing. And this increase is very dramatic. And so the concern is obesity is not a single disease, as it has been demonstrated earlier. It correlates with risk of cancer in multiple tissues, not limited to colorectal cancer. Colorectal cancer is one of them, but there are different tissue types that have significant association with risk of obesity, and some of them share some similar molecular or developmental origins, but some of them do not. So that is a very interesting question as a basic scientist to me to understand how does obesity increases risk of cancer. So there are multiple paradigms that we can use to explain this phenomenon. One is that we know that obesity has this epidemiological link with cancer, and there are some association studies that hint certain causal molecular mechanisms, but we really don't quite yet understand how does diet directly or indirectly influences risk of cancer, dependent or independent of state of obesity. So I just wanted to highlight that this is, again, a very complicated problem with lack of causality and mechanistic studies. So we did a modeling study when I was back in Boston, where we fed normal laboratory mice with a long-term high-fat diet. This was at least for 9 to 12 months without any mutation or carcinogen. These mice, at the end of the cohort study, were seen to develop more significant carcinoma and low-grade dysplasia when they were fed with a high-fat diet. So that's in normal laboratory mice. If you age them for 24 months, this is something we knew. They developed tumors in various types of organs, and in black 6 mice, that was the association that we have done. So when you give high-fat diet as an input, which ended up forming obesity, we get more tumor as an output. So in terms of the mechanistic studies, one needs to be reductionist, because there are, again, hundreds of potential pathways that are perturbed. So we took an approach to look at the initiation events for many reasons. As in the clinic, you would know the association between how many polyps you may get may correlate with later on how many tumors you may end up having. So this is the model that we used. We focused on the stem cell compartment within the intestinal epithelium and developed certain models, organoid models or animal models, to demonstrate that in the context of high-fat diet, how fatty acids can lead to abnormal stem cell activity and how this abnormal stem cell activity may contribute to risk of cancer. So I'll summarize briefly here. In normal intestine, your stem cells can self-renew and differentiate to give rise to cells of intestinal epithelium, and then they can interact with nutrients at steady state for homeostatic conditions. And most mouse model studies definitely have shown that is the oncogenic mutation that can occur within the stem cell compartment can contribute to the tumor initiation events much more than the oncogenic events that occur in differentiated cells. And it's due to the cycling state of the intestinal epithelium and the fact that differentiated cells most of the time will fall off the next day through anucleosis. But in high-fat diet, what we found was these fatty acids activated this lipid-sensing transcription factor called PPAR delta, which has been implicated in colorectal cancer for many, many years, and there were conflicting reports in mouse models and so on. But we found that fatty acid-induced activation of this transcription factor and its coupling with beta-catenin, which is the main responsible transcription factor for stem cell maintenance and also the highest implicated transcription factor in colorectal cancer. So when APC is mutated, you get more beta-catenin activity. So we found that link within the stem cell compartment, but also we found that in non-stem cell transit amplifying cells. And now with single cell analysis, we are seeing overall in the crypt, we see this elevation, augmentation of beta-catenin transcriptional activity. So that was a causal mechanism that we were able to demonstrate that link high-fat diet or the high-fat diet-induced obesity to intestinal cancer and opened a paradigm in which where we can potentially target PPAR delta in the context of obesity-associated or high-fat diet-associated cancer types. So we published this 3 years ago now, and later on several other papers demonstrated a similar paradigm with fatty acid utilization or PPAR activation in the context of prostate and head and neck cancer progression and metastasis. So this is not limited to the colorectal cancer risk, but this lipid utilization or lipid-induced activation or augmentation of PPAR and beta-catenin may be a contributing factor for risk of cancer in different tissues and states. But to me, that is just one part of the puzzle and a very small part of the puzzle, because when we take those nutrients or when we think of obesity, it's not altering only epithelial biology. And when we look at the intestine, this is a very simplistic cartoon model. Epithelial barrier is right here, but there are multiple other modules that interact with these epithelial layer, which can contribute to risk of cancer. So those are immune modules, gut, microbes, and metabolites, and so on. And also neurons are implicated in that. And then we need to see this through a holistic manner through the organ to understand how the organismal physiology may contribute to that. So I will just briefly summarize the model and the data, how we look into this now. We have put our attention into how immune recognition mechanisms and immune surveillance mechanisms may be impaired or altered in obesity, and risk of cancer may be influenced by that. So there's a long literature in understanding how tumor-immune interaction occurs. And so I will show you briefly how tumor immune evasion may contribute to risk of colorectal cancer in the context of diet-induced obesity. So CD4 and CD8 T cells are the cells of adaptive immunity. They produce cytokines or cytotoxic molecules that can target the tumor. So there's an orchestrated, integrated immune response to tumor. it's not just like one cell type that fights the tumor. Most of the time, we hear about CD8 T cells, but it is an orchestration of all these different players that actually can give an effective immune response to the tumor. So tumor usually downregulates immune recognition molecules to escape from that. So MHC class 1 is a main hit in that context, but also MHC class 2 downregulation has been seen in certain types of tumors that express MHC class 2. And so what we found is in intestinal epithelium, there is very high expression of MHC class 2, and so it is significantly downregulated in high-fat diet. You can see it here. So we wanted to test how this downregulation of MHC class 2 influences the risk of cancer, and then we did a mouse model, a mini colonoscopy system where we can inject the tumor-initiating cells into the distal colon. And here you are seeing when you inject premalignant cells that can form tumor, if they don't have MHC class 2, they give rise to many more tumors compared to MHC2 intact counterparts. So this is in immune-competent host. In immune-deficient host, there is no difference, suggesting that immune surveillance is an important mechanism. And we found that this is most likely due to the microbiome alterations, because in the high-fat diet and in antibiotic treatment, we have, and others have demonstrated that there is dysbiosis. And then here you can see, okay.
We turned it off.
Oh, okay. Maybe this one. Well, you can see that when you get rid of all the microbes, the depletion of the microbiome correlates with the reduction in MHC expression pattern. And I'm not going to show you the data, but we have very convincing causal data that bacterial-induced pattern recognition signaling and cytokine signaling maintains MHC expression, and these alterations can contribute to risk of cancer. So in summary, in normal dietary situations, in the intestine, tumor-initiating cells can be surveyed by immune cells through MHC class 2. And in high-fat diet, we see the downregulation of class 2, which leads to impaired immune recognition, and the tumor can be formed more effectively. And this is correlated with the state of the microbiome, which we are now trying to determine the causal microbes and what are they producing and how they are positioned in the intestine. So in summary, I just want to highlight the necessity to integrate multiple modules and have a unifying theory of the risk factors for colorectal cancer. I showed you initially that intestinal stem cell-centric view can help us explain certain things, but most importantly, how the nutrients, different nutritional profiles. Here we use just a single type of high-fat diet. Now we are expanding our dietary cohorts into different types of dietary regimens, and also the T cell or immune cell, cancer cell crosstalk, how they are directly being perturbated by the nutrients, and also due to microbial alterations are very important. I will give you some vignettes of the phenotypes we are studying. So there are 4 of those. So 2 of them you have seen the data for, but the next 2 are about immune recruitment, which is very important and has been discussed in cancer immunology field extensively, why certain patients respond to immunotherapy and the others do not, although they share all these molecular proxies. So immune recruitment, I think, is a very important aspect. And the immune cell response, not just in terms of the checkpoint mechanism, but the fitness of the immune cell. Are they metabolically capable to proliferate and attack? So those are the 2 other recent observations that we have been making. And I think that's the end. Yeah, thank you so much.